I have an honor and pleasure to invite Professor Jay Dean from South Philadelphia University, and with the state-of-the-art lab and a bunch of very good people in his lab, he will try to answer the very interesting question, is it possible to prevent CNS oxygen toxicity?
Thank you. Thank you for the nice introduction. Thank you for the invitation. My first trip here, I've been enjoying it. All right, the question, the title here is Prevention of CNS Oxygen Toxicity. Well, not yet. But what I would like to go over are some new thoughts about how we might be able to predict seizure and some pre-dive strategies that will delay seizures. And hopefully this is the start of something that will ultimately allow us to maybe even prevent seizures, but we'll see. An outline, first thing I'm going to briefly mention is a brief tribute to an electrophysiologist extraordinaire, Dr. Grossman. I'll then get into the research and the data, and the first topic is hyperoxic hyperventilation, which we're proposing is a physiological marker or a physiomarker that will predict an impending CNS oxygen toxicity seizure. This story will have two parts. Initially, I'll go through some of the in vitro brain slice studies we did, where we were able to identify that CO2 sensitive neurons in the medulla, cells which we think are chemoreceptors for control of breathing, are also very sensitive to hyperbaric oxygen. And this led us down the path to the hypothesis that we might see early changes in breathing that precede seizure. And so to look at that, we used hyperbaric radiotelemetry, so we could work with untethered, awake, unanesthetized animals and look at their breathing and their cardiovascular responses to early exposure to hyperbaric oxygen. Then I'll move on to another topic, pre-dive conditioning or preconditioning, where intermittent exposures are given every other day for about an hour using various gas mixtures over a 5-day period. There are sublethal exposures so that we don't cause seizures. We'll use a low level of hyperbaric oxygen, and then we dive the animal to 5 atmospheres to see if perhaps by induction of reactive oxygen species we've upregulated endogenous antioxidants to block seizure or to delay seizure. And so we used oxidative preconditioning. We did hyperbaric oxygen. We also tried hypobaric, hypoxia. And as I said, these were sublethal doses that we used. And then we did some control experiments where we used normoxic gases to look at, to rule out any effect of pressure. And quite serendipitously, what we discovered was that there was a pressure effect, and we did get some barometric preconditioning, so I'll go through that. And then the final topic will be a brief mention, which will really introduce the next talk by Dr. D'Agostino, of how pre-treatment with a ketone ester 30 minutes before a dive can essentially mimic the blood chemistry of being on the ketogenic diet for a couple of weeks, and it has a very strong, significant effect on delaying oxygen toxicity. So initially here, I got this photograph from Raffaele, and what struck me about this picture was the little vibrissae sticking out here, and it reminded me of another picture I'd seen earlier, and oh yeah, there you go, you can see they're related, I think, but I'm sorry, Rami, I couldn't resist. But just as a tribute, I'll run through this real quick. Too fast for you to read. He just published too many papers, Rami, so but this is what I could find that he had contributed to our literature on pressure. And if you'll look there, you'll see everything from studies of respiratory control to synaptic efficiency and so forth. So thank you for all that you've done for us and helped us. All right, so this story will start with a discussion of CO2 chemoreceptors. It's something I've worked on since I was a postdoctoral fellow. And just as a brief review, CO2 chemoreceptors are distributed in several sites in the medulla. And if you start here in the brainstem, you can look at the area that we're interested in, which is the solitary complex made up of the nucleus tractus solitarius and the dorsomotor nucleus of vagus. Some people call it the dorsal vagal complex. It's an important area. It does a lot of things. It gets the afferents coming in from the peripheral chemoreceptors. And then it makes synaptic contacts with other chemosensitive areas down here in the retrotrapezoid nucleus in the classical area, identified back in the 60s, the ventrolateral medulla. You've also got the medullary raphe and locus coeruleus up in the pons, and of course there's some other sites which don't show up too well on this picture, but it's a distributed system. I'm going to focus on the solitary complex here. And if we look at this, this is an area that is known to be CO2 sensitive or chemosensitive. In fact, all those areas we just looked at have been identified using this method where they will place a microdialysis probe down into the nucleus region. They'll dialyze in a CO2 saturated solution. They're using 25% CO2 here, but what they've measured with pH probes is a very mild focal acidosis of about 0.01 to 0.02 units that will cause a local acidification, presumably stimulating central chemoreceptors, and you can see that you get an increase in breathing. And there's an interesting relationship here. Some areas are more active when the animal's awake, some are more active when the animal's asleep. In the nucleus tractus solitarius, which is part of the solitary complex, you can activate breathing during both sleep and wakefulness. So if we go back to when I was a postdoc many years ago, and we'd make a brain slice through that area, got about a 400 micron thick section of tissue, and we bathe it with an artificial cerebrospinal fluid, and you go in and you can record from neurons located here and here. And this is just extracellular recording of integrated firing rate. And you can see as we go from we're going from a 26 millimolar bicarbonate solution with 5% CO2, which gives you a pH of about 7.4 in the medium, about 7.2 in the cells. And we raise it from 5 to 10%. You can see firing rate goes up. If you then superfuse the slice with high magnesium, low calcium to block synaptic input, you can see you have some modulation here synaptically, but the cell still responds to CO2, so it's intrinsically CO2 sensitive. Okay, so we thought that these were good candidates for central chemoreceptors, and the fact that you could evoke a respiratory response from that area kind of supported that hypothesis. Well, I got interested, as I said last night at the party, in working with gases under pressure in the early 90s. And the system we came up with here initially is shown, which actually worked quite well. You could push the sled in, and you could leave the door open and poke around with a sharp electrode and get a recording, and then actually even close the door, close the yoke on this without losing the recording. So it became very useful. You could keep the door open, keep changing electrodes as often as you had to. And using this system, what we found, we said, well, let's go right into the solitary complex and see if the cells respond to hyperbaric oxygen. And lo and behold, they did. So here we are going to 3 atmospheres with helium. Once we're there, what we did was we would dissolve oxygen in the ACSF in a separate pressure vessel and then perfuse that into the chamber so that we never pressurized the main chamber with oxygen. The oxygen was all dissolved in the medium. And so you can get a nice reversible, repeatable stimulation of firing rate here, and conductance changes. You normally see input resistance going up or membrane conductance going down. And so when we got these recordings, we said, well, let's change CO2 and rule out the central chemoreceptors. Well, as it turned out, that was the population of cells that was most sensitive to the hyperbaric oxygen. And what we found was that 90% of the HBO excited neurons were also CO2 excited neurons, or the central chemoreceptors that we'd been studying all along. And if a cell was CO2 insensitive, it usually was not sensitive to hyperbaric oxygen. So we started playing around, pairing the stimuli together. You can see here's a cell being depolarized. This is integrated firing rate here. This is membrane potential. And so we're at 1 atmosphere. We give it high CO2 and stimulates. We now go to pressure, and we're using the same CO2 stimulus here. We've adjusted the fractional concentration of CO2 under pressure. And you can see the CO2 response again. And now when you pair it with HBO, you get a very strong stimulation. And then just another example, here's a HBO stimulation of firing rate. HBO stimulates again, CO2 gives you a big response. And then here's CO2 by itself at 3 atmospheres. So And I won't go into it, but we think there's some interactions here where acidosis can drive the Fenton reaction, and where CO2 can react with peroxynitrite to amplify some of the ROS and RNS production. So there appears to be a very powerful effect on these neurons of both oxidation, shown on the left side here, and of pH coming in on the right side. Now, I don't have time to go through all these data, but this is what we've established. What we know about these cells, they tend to acidify. They remain acidified when they're exposed to CO2. Extracellular acidosis inhibits the sodium-hydrogen exchanger here, so they don't show any pH recovery. So that's a good sign for a chemoreceptor. Chemical oxidants will also cause pH to drop, and so they're both, what we've seen electrically is that they both increase input resistance or reduce membrane conductance, which most likely is a potassium channel. Firing rate goes up, and you can see we proposed increased ventilation. Well, is there evidence of that? Well, yes, there is. And so we're saying that both hyperoxia and pH and the ROS that's made from hyperoxia are what set the activity of these cells and thus your level of breathing. So there is in the literature a paradox. We all know that hypoxia stimulates breathing. Well, hyperoxia also stimulates breathing. And so this is a human adult here. This is a study done where they are going from air to 30% oxygen to 50% and to 75% oxygen. You can see there's a strong stimulation of breathing. It actually is a potentiation even afterwards, after they've removed it. And they found that this response was biggest if they used what they called isocapnic hyperoxia. Because what we're going to see is, as you stimulate breathing with hyperoxia, you actually begin to blow off CO2, and you can blunt the magnitude of the ventilatory response. And so if they titrate in a little bit of CO2 to keep end tidal CO2 constant, then you see a very strong stimulatory response to breathing. So this has been seen in adults, also been seen in infants, and in experimental studies in rat. This was an anesthetized rat, and you can see they gave them, let's do this here, 4, 6, and, whoop, they're going too fast. The solid lines 4, 6, and 8 atmospheres, stimulated breathing. And then what they did was that they cut the carotid body afferents to remove the peripheral chemoreceptors from the loop. And they asked the question, is this a central response just due to central chemoreceptors? And what you see at 4 and 6 atmospheres is the hypercapnic ventilatory, or the hyperoxic ventilatory response is retained. 8 atmospheres, it was blunted, probably due to a more of a CNS toxic effect there. So this has been repeated in other labs. So the fact that we found intrinsically oxygen-sensitive cells that were also CO2 sensitive, and the fact that hyperoxia can stimulate breathing, we began to wonder if what we were seeing in our in vitro studies could also be seen under in vivo conditions. So this is kind of summarizes, this is a slide that we shared with our funding source, ONR. And it's showing that when we do our brain slice experiments, record from cells, we find oxygen-sensitive neurons. And what we've also found is that when we measure reactive oxygen species, in this case superoxide, we find that in the solitary complex, you see a very high level of ROS production compared to another area like the hippocampus. So the fact that there's this high level of ROS production and this high oxygen sensitive electrically, we began to wonder if, because of this, would we be able to see a predictable pattern of early changes in cardiorespiratory that precede onset of seizure in HBO? So we're not saying that the solitary complex neurons are causing the seizures, but the fact that they're so sensitive to oxygen, perhaps once the dose of oxygen reaches a critical level, it starts to be dangerous and will cause seizures, we'll see a change in the breathing pattern. So, and in fact, I should say, there were anecdotal reports in the literature where divers, Chris Lambertson, who had mentioned that just before he had oxygen toxicity seizures, he was conscious of his diaphragm having spasmodic contractions. So we thought we would look at this then by using radiotelemetry in animals and to see if this whole animal response, in this case you can see breathing starting to increase here during HBO before the seizure, and you'll see that's the trend that we did find. And so ultimately we think that our cells that we've been studying underlie this response that I'm going to be showing you under in vivo conditions. So the approach is to use a 4-channel dual module transmitter. It's a battery pack here, and this is the telemetry unit. When we implant electrodes into the head, into the chest for EKG, and into the diaphragm. And we actually had some problems early on. The software did not sample fast enough to give you tidal volumes, so you couldn't look at minute ventilation. And so we worked with the company to get a higher sampling rate. And this is just showing early on, if we, here's the wires coming in here's the diaphragm. We were implanting them right into the wall where the diaphragm inserts. So it's kind of a mix of diaphragm and these abdominal muscles here. And we wanted to see how the electrical signal compared to what we would get with the plethysmograph. And you can see we get a very good correlation here of the diaphragmatic EMG to the actual flow in the plethysmograph. So we got a good measure of breathing. We wanted to make sure that the radio transmitters were not pressure sensitive, so we made up an auger analog here. And you can see we did EKG and diaphragmatic EMG and EEG. And what we found using stimulating electrodes here to give us signals to look at, we found no effect to pressure. And so these are pressure insensitive, which made us happy. And so here we go. Raffaele showed you this slide yesterday. The receiver is beneath the animal, and we are perfusing or flowing 5 atmospheres of oxygen, either air or 1 atmosphere oxygen or 5 atmospheres through here, and then in parallel compressing this with either air or nitrogen. And you can see we don't get any CO2 buildup into the chamber while we're doing this.